Hello.
Hi, how are you?
Good.
Hi,
Hi, Victoria.
Victoria. You have a new picture. Nice.
I keep changing. Yeah, we went out to sushi last night.
Oh, nice. That looks like a fun place.
It was good. We came out in a big sushi boat.
Oh, really?
It's crazy.
Hi, Victoria. Oh, I said already. Hi, Victoria. I'm so sorry. I'm a little bit. Yeah, welcome everyone to the weekly recap. This is the room where if you missed any of our rooms during the week with the guest speakers, or if you just want to have a quick reminder, that's what we do here, usually on Sundays at this time. And then if you like what you hear, we just do a short version. Then you can either go back to the full replays and listen to the whole story. So, yeah, thanks for coming. Hi, Jamie. How are you today?
Hello, Katerina. Hello, Science Society. I am well today, thank you. Have you had a good day so far?
Yep. Uh, yeah, I've unpacked a lot. There's still a lot, but
Ha!
I
How
showed
close? How
Serena
close?
my progress. You can ask Serena for grading what I
See,
did.
I've been here a year. There's still stuff I haven't unpacked, and she's like got the whole house set up. It's crazy.
Really? Wow, I am genuinely impressed.
Yeah, I couldn't possibly.
Well, thank you. I'm exhausted.
How do you do it, Katerina? Are you using this renewable energy that some of our speakers have spoke about? Because you're not using the kind of fossil fuel consumption that I'm doing.
Well, funny that you say that because Serena and I, we were talking yesterday. Once Science Society makes like a major event, we will of course get approached by designers like Valentino and so on, that they will make gowns for us for the castle
Yes,
event.
absolutely.
But
Yeah, I
but
want a ball
our
gown.
yeah, but our requirement is that it has to be made out of smart materials that while we walk, we create carbon capture through the friction. That's the
Oh,
least.
I love this. I love this idea.
This is
Carbon capture through fashion.
yeah, the carbon negative ball gown.
This is going to be a thing. Yeah, in fact, put even the carbon capture at various places in the gown so it gets like, does it get darker over time as it captures carbon? Would it change color?
That would be really cool, right, Serena?
Oh yeah, it
The
it
more you
it
move.
fades to black.
Yeah, so implement that in the design then, right? So as it captures more and more, it's like creating this, I don't know, a picture or a symbol or something, or maybe not a symbol. But yeah, no, I can see that working. I can see fashion people, designers going crazy on that.
And then the cool thing is it motivates you to move and burn calories, right? Because you would capture more, so you get a higher score.
Right, so
Oh
if you
yeah.
wear a ball gown, get prepared to start dancing.
Yep.
Catch that carbon. Save the environment, Serena. Get dancing.
So,
One tango at a time.
Well, yeah, there could
so
be
Valentino, if you guys are listening, that's our requirement for wearing a ball gown at our castle. So,
They'll capture a niche market, the science folks and the carbon neutral folks.
Absolutely.
maybe we can think about if they apply for being our in-house designers. We can think about it if they come up with a solution like. Anyways, joke aside. Let's start with the recap again. Thank you everyone for coming, and I hope you enjoyed this last week with us. So we had on Monday Dr. Jyoti Chatterjee. He is at the University of Strasbourg, a researcher, and he investigates the molecular mechanism of transcription-associated proteins contributes to our long-term memory. And currently he is in Ted Abel's lab at and Ted Abel, he is the director of the Neuroscience Institute chair and DO Department of Neuroscience. So he's the senior scientist of this project. And yeah, if you look at the presentation that he shared with us or he made for us, so what he published was the molecular mechanisms of long-term memory, how long-term memory basically gets stored in the neurons and what goes wrong, especially in Alzheimer's disease and related dementia diseases. And it's a really interesting mechanism and very important to know this mechanism that basically CREP binding proteins, which is a very common mechanism, like it's a pretty, so whenever any major excitation goes on in neurons, this CREP gets activated and then genes will be expressed that basically will implement that activity in the neurons, and if they are strong enough they will get stored long term. And he dissected out the really detailed mechanism. He looked at it in different ways, through through gene expression mechanisms, and yeah, the so if you look at the paper, he then later on looked at what the CBP pathway is, like how the CBP pathway works, and then if there is a potential therapeutic target in Alzheimer's disease mouse model. And he then saw that NR4A genes are progressively downregulated across increasing grades of Alzheimer's pathology, and he then used drug that targets this pathway, and he could basically rescue the capability of memory consolidation. So this will have huge implications. The knowledge he gained for neurodegenerative disorders. I'm not sure if anyone has something to add for this talk.
Well, for when this seemed like it was ages ago. I can't believe that was just Monday. But I was curious that there, because I kind of thought that more, astrocytes were more involved, but he gave a complete neuron only mechanism. But it is, in terms of the signaling cascade, I think the astrocytes are involved upstream from that, but I wasn't sure. But this was cool. These were in the hippocampus, right? So this is where the genetic expression is altered and the receptors are upregulated. But I did think there was astrocyte involvement in that. But,
Yeah, yeah, exactly. So they did this for the hippocampus, but he said that this NR4A pathway and is not unique to hippocampus neurons, and in the future it would be really interesting to look into more brain regions. And he didn't study the astrocyte involvement, but yeah, he says that there is for sure a regulating factor from the astrocyte side. But he didn't study it yet, but it would be interesting in the future.
I found it really interesting when he actually was talking about the spatial memory being located in the hippocampus, and that's where he was kind of working on, right? And it was, to me, it was mentioned a lot of things indicating, why we find things like memory palaces and stuff so effective, why, geographically remembering things is kind of easier for us because it's a common like held memory technique, right? Um, and it made a lot of sense to me when long-term memory and memory storage was happening in this place involving spatial awareness. Um, Found that incredibly interesting. And then hearing the biology of what the neuroscience to what was happening when memories were actually being consolidated. Um, he had mentioned here that to convert a single memory trace of sensory information into stable long-term memory um, requires a post-experience process called consolidation, which, to put it that way, doesn't sound very impressive. Like after you learn something, you need a bit of time to sort of think on it. But it was fascinating when the doctor was actually describing the actual procedure that was happening, right? When he was talking about the protein folding and stuff like that, wasn't it, Katerina? It was just incredibly interesting to hear the biological mechanisms happening to why consolidation is important and what it's doing as well. That was my interesting take, or one of them from this amazing talk.
Yeah, didn't it? I mean, it was like if you really want to teach kids stuff, you it would imply that, which makes sense. You give them, some lessons, but then you gotta like have downtime for them to consolidate instead of just ramming it one class after the next or something.
Yeah, yeah, I agree that I think a lot of the ADHD problems we see nowadays are just a reflection of how we completely keep overstimulating during development the brain, I think. And this just confirms the theories I had before quite quite specifically. So I agree.
And definitely discussing the mechanisms behind why consolidation is important is probably understated and should be spoke about more. Because otherwise, when you tell people a kid or even an adult needs a bit of time to process, it somehow sounds almost lazy. Like, oh, I need time to consolidate. Yeah, yeah, yeah. Get back to the next class or the next lesson or the next section. It's not something you can hold on to. It's just someone saying, I need time to consolidate. But hearing the biological factors. Like, no one would argue if you work really hard in the gym that your muscles are really, really sore and you can't just go and start lifting more. Nobody would argue on that front. But understanding better what's biologically happening in the brain when memories are becoming memories, um, maybe that would get the same amount of respect.
Yeah, I think it justifies my afternoon naps.
Yeah, let's
Someday
go with that, Serena. Yeah.
I need to consolidate.
But remember, I actually did ask him if small naps after an event was actually beneficial. He did say he couldn't speak to the efficacy because he doesn't really know. But he did mention about sleep being important in general for um, like memory creation and keeping it quite what was it called? Accurate. What was it called? I forget the term he was using, but making it a very good memory though. Sleep was very, very important for that.
Well, let's all experiment and have, when we read papers, have naps.
I'm down for this experiment. I will try this.
Okay, shall we
Oh,
move to the next one? Or yeah, go ahead.
Oh yeah,
oh
we
yeah.
can move. Go ahead.
Oh no, I was just going to quickly last say that I was really interesting what he was saying about the tests he was doing with the mice, how he used was it special object trace, I think he called it, with the mice when he put them out and he put three objects down and he let them examine this and then he put them back into their cages where he gave them like minimal stimulation so that they could consolidate. Then he removed one of the objects, and he was noticing how the new object was something that caused it to notice it more, and it was actually always studying the new object more, which he was indicating that new changes to what they remember causes them to pay more attention to the changes. I actually found that really interesting. Sorry, that was my thought.
Okay, I'm trying to pin the link, but my app didn't, it started freezing. I had to restart it, so now it works. Okay, good. Sorry about lately the app has been freezing. Okay, so we had Dr. Garin as the next guest speaker. He talked about the human brain evolution of abstract thought, and he's at the Vanderbilt University. Yeah, this was also a really interesting talk. So let's go over it. Yeah. So he mostly, so he looked at this evolutionary gap in the primate default mode network organization. So he looked at the diversity, first of all, the diversity of different architectures, size, weight, and then numbers of neurons, and so on. And based on that, he did. He went over, all this basic about different states during disease and um, how the brain activity looks, and then he looked at a resting state network in different mammals. And he found basically a really quite interesting difference between humans and other mammals in this DMN, the default mode network. It was a lot of brain imaging data that he shared with us, which is really impressive to go and have the ability to look at different sized mammals. So he looked at human, macaque, marmoset, and also lemur. And he saw that in non-human species, the medial prefrontal cortex involvement is really minor in this default mode network, and there's really a different, distinct non-human DMN that he dissected out. And later on, he even shared data sets that are not published yet, where he basically looked at a lot of data from different mammals again and saw that it's not really a completely switch as they expected before, that basically all of a sudden you have human primates and the network just switches to become very different. It's actually a slow tendency that grew over time or changed over time to use this DMN that we see in humans. If you look at slide number 25, that displays really well that evolutionary history that evolves towards this default mode network that are seen in humans. So it's not like a completely new way of thinking. It's just evolution that slowly evolved towards this network structure. And I think Serena asked some Glia questions about that, if I'm not
Surprise. Yeah, no, it was really interesting that just how different the default mode patterning was in humans versus the other species. It's like I don't know how what the deeper implications of that would be in terms of some of the models, animal models that we have. But it was interesting that just completely different regions are used when in the default mode. That was kind of fascinating. But their data was amazing. They had a much stronger magnet. What were you going to say, Katerina?
Oh yeah, because of that, we mentioned that mental health drugs don't work really well, and then if it's higher thought, let's say worry of the future that humans have, um, if we test this basically in animal models that have a very different network, the brain regions they use. The question is that the problem why our mental health drugs don't do a good job in helping people? So this work is really important. It wasn't done for helping people with mental health disorders per se. It was just done out of curiosity, how brain networks change throughout evolution. And then I think it becomes this huge, important groundwork for changing on how we hopefully will develop mental health drugs in the future, taking this into consideration. Hopefully, this is not always the case, right? Researchers find things and people completely ignore it for decades until somebody picks it up. So yeah, we have to make a good job in putting this knowledge out there as much as we can, because I think this is really crucial for developing. I just heard yesterday a podcast about how much suicide rates are in children increased the last years. I think every year around 400%, and the kids are getting smaller and smaller. Like I think one 6-year-old was last year the one of the younger ones, but it's not uncommon anymore that a 6-year-old commits suicide. And I think and with the mental health issues increasing, we have to really do a better job. And I think this shows how just really basic curiosity research is really important, because you don't it's to solve neuroscience, it's a very complicated field, and if we do it in a planned way, I'm doing this to solve this, most of the time, like not most of the time, but many times, not much useful stuff comes out, but all of a sudden just a pure curiosity science project solves this, because it's so complicated. We don't do a good job at planning exactly the experiments needed. So yeah, I just wanted to put that out there.
So, Katerina, are you driving at like the difference between systematic studies and hypothesis-driven work?
Applied versus basic research.
Yeah, I've been thinking about this a lot. The need to do things more systematically rather than just chasing down one hypothesis at a time. But yeah, just to add maybe just a little different perspective on this, because I saw the talk and it was really, really interesting. It overlaps a lot with, I've been thinking about comparison across species a lot lately. It's really interesting. It's always fair game to apply the same method across, different species or different conditions, and then, report what the differences we see, and that the results are interesting. Um, I was recently reading this paper talking about the default mode network. Apparently, there's an exponential increase in the use of the words default mode network in published papers right now. It's like exploding. But to inject the other viewpoint here, there's a big question about whether this is actually means anything. Like, it's not very clear that the idea of a baseline state for the brain actually makes sense or means something. And, putting aside all the kind of quibbles across the field on this kind of stuff about what's included in the network and what's not, but a lot of the studies that try to get at what the functional role of this might be are really, really difficult to interpret, right? It's like lesions and things like this. It's hard to tell. It's not like we have a manipulation that gives us like a direct effect on whatever the function of this is. It's likely disturbing a lot of other related processes. But yeah, anyway, it was really interesting, really well done studies, really, really cool talk. But I thought I would add in maybe the other opinion on the default mode network stuff in the field.
Yeah, it'd be great to see how just within humans the variation across a much broader sampling to see how much it holds up, and with a diversity of personalities, what they do in their resting state and how their brain operates. Is there general structural correspondence or regional correspondence, or just is it as various as what people think about when they're just resting?
Well, the thing is, what I was saying is that, Joseph Ledoux has been writing about this for the last 3 years, that all we did for anxiety disorders research in his lab and in other labs came up with these different treatments and drug treatments that don't work. They basically downregulate the excitability of the brain, but that's it. Real anxiety, like an immediate threat response, decreases. But this anxiety that you have, fear about the future and different scenarios that you come up with in your mind, they don't get addressed by any of the treatments, and especially the drug treatments. And he wrote that the problem is that we are doing all these tests in other mammals that don't have this complex thought process for the future worry type of processes. And we cannot really translate what we do in rats for very complex fear of the future, what could happen to us that happens in humans. And this data set, before we didn't have real factual data set, he wrote this, and he annoyed a lot of people with this, because it's basically 20 or 30 years of research. He says you can all go home with it. Not as strongly, but kind of that was the message. And this is the first real data set. If it's completely 100% accurate and stuff, yeah, that is debatable. But this is at least the first data set that shows that this could be the problem. Why stuff is not working, how are we doing it? And why for certain things we cannot use animal models to study it. It just doesn't work.
Yeah, and reading about the astrocytes' role in binding emotion to memories, and what you actually feel, or how the moment actually feels, or memories how they feel. The binding between the amygdala and the hippocampus, is has a strong astrocytic coupling to it. I'm wondering though if are the is the drug discovery efforts for anxiety in these in other areas actually targeting that process, and PTSD treatments and so forth, because the emotional coloring of memories, um, it strongly involves the astrocytes. So this is just another angle. I wonder if what we're seeing in the data, though, because the metabolism of the astrocytes and how they connect directly to the vasculature, and they actually synthesize glycogen, and that glycogen is broken down into lactate that's fed to the neurons for their energy supply. I'm wondering if what we're actually seeing in the data has a if we're looking to some larger extent at the metabolic activity of the astrocytes and how they're regulating the blood flow, and and supporting the neurons as they fire, which is kind of interesting in its sense. It's sort of a reinterpretation of that data.
Yeah, I think I agree. I think that's a really interesting point. And what I also wonder, the thought just came to me, and it's probably nonsense because I would read more, I would need to read more about it. But don't you think that maybe anesthesias um, target more astrocytes than anything else because of what they mostly do? They disrupt the large scale communication between brain regions. So that's how they kind of disrupt um, consciousness. Like there's still brain activity, it's just very separated. It doesn't oscillate throughout the brain. So maybe it would be interesting to maybe use low dosages of anesthetics. And the interesting thing is we are doing that already with ketamine. Ketamine works really well for depression and severe anxiety disorders that are treatment resistant. So I'm wondering if it actually has a biggest effect on astrocytes.
I would certainly think so. It's a hypothesis that I would really be interested in seeing more pursued, in the sense that the strength of these calcium waves. So all of essentially the major neurotransmitter receptors are expressed in these astrocytes there. But I don't know how many drug treatment efforts or therapeutic treatments really study them, and in the sense that the that they are that's the network where it's actually coordinating the neural synchrony and the synchronous firing, and is implicated in these higher cognitive functions. I think we really ought to give it a lot more attention when we're looking at therapies. Um, but
I think the only way forward, I don't know, but again, the real mechanisms, when we look at the actual mechanisms, how ketamine works molecular and on the neural network in that region is in animals, but we know that astrocytes in humans are quite different. So I think the way to go would be to create pretty good organoids with a pretty accurate ratio of astrocytes versus neurons, and then look at the mechanisms how these drugs work in organoids. So,
oh yeah, when we get to the organoid, when I'm going to have some more fun. That was a great talk.
yeah. So yeah, let's go on to Dr. Moultrie.
Oh, are we already to Motri, or we just skipped?
Oh, we skipped somebody? I don't
I'm not
think
sure.
so. Moultrie is the brainwaves, then it's Zuniga about
Okay,
the
cool.
Tauta piece, and then Henry, Dr. Henry was. Yeah, so Dr. Allison Moultrie, he is at the University of California, San Diego School of Medicine, and he's the professor there, and he achieved many awards. One is being the rock star scientist, and I think he totally earned that. He completely earned this, and it was like a really amazing talk. It was like epic. It was an epic talk. So what he does is he creates organoids of human brain tissue, basically a human brain, a mini human brain in a dish. And he created this. At first, he talked about how they created a pretty good organoid recipe. It has kind of similar gene expression patterns as in regular human brain tissue, as far as we know. And then they can grow them pretty well and actually keep them. I think he said up to 3 years was the longest one they kept brain organoid. And they then started looking at some point at the organoid network activity and also long-term network activity, and it had some neural correlates to a regular human cognition, and network oscillations emerged out of this cortical organoid network, and it became it mimicked the bursting phenomenon of an EEG of a very small baby, of a preterm baby, and yeah, they could really record LFP features that were quite comparable, and this was really interesting. The next thing, he then moved on to present unpublished data, like real ongoing research that he didn't share before, at least like in this type of setting, probably in lab meetings and stuff. But
So you heard it
with
first here on Science Society. Yeah, there we go. Yeah, Katerina, please.
exactly, so they want to add more sensory stimulation to the organoid. So what they did, they created an embodiment as a little robot with different sensory inputs, and um, and connect that to the organoids, which is amazing. So you have basically organoid that has its own little robot, and they will observe now how well that works, and it will, if it will make the organoid mature even faster or better or have different features, how the organoid will control that robot. So that is coming up, and he promised to come back. Um, he also develops organoids for different mental health disorders and things like autism, spectrum disorder. The other project that is still ongoing and not published yet is he also uses them to study evolution of humanoids. That was another really interesting study, and it kind of related to what we heard in the talk the day before. Um, and yeah, so he looked at different genes that may have resulted in a more sophisticated brain, especially a few genes that he saw that were related to neurodevelopment, and mental health disorders were changed throughout evolution. So he reconstructed the ancestral brain basically, and created ancestral brain organoids. Yeah, he shared so many things. So he showed a distinct neurodevelopment, that the maturation was earlier in ancestral organoids, and there was also altered network neural connectivity. And yeah, they are still working on it in future experiments. And then they are also sending organoids out in outer space. They won a NASA award for $5 million to basically see what the effect is on this organoids in outer space. So they want to basically create and see how faster aging affects these organoids, to study aging. So yeah, they are doing a lot of really exciting stuff. Check out their website. In the end, they have the page 41. You can see they have a stem cell channel that also discuss about stem cell ethics. He's very concerned about ethics with this organoid studies. And yeah, check out their channel and keep checking out this website. He, I think, updates pretty well his current research, and it's really stunning. It's really awesome.
May I just say that if for anybody listening to this right now, there's something here for everybody. If you're a biologist or a neuroscientist, there's plenty of organoid action going on there. If you're actually interested in robots and little cybernetic organoid robots, there's something for you there. Even if you're a science fiction writer who needs some ideas, come to this replay and listen to the amazing things he's doing, because it was giving me plenty of ideas I was scribbling down as well.
Yeah, this was just an absolutely amazing body of work. Um, Certainly, and when we get talking about astrocytes and the developing these organoids, what was really fascinating is that there was, non-coordinated neural activity until it was about 2 months, and they just sort of came online and started firing together in synchronicity. And I asked him, well, so what were the astrocytes doing? And it was very clear to him, yeah, that's when the astrocytes sort of came online and started driving this synchronous behavior. And it continued to, the actual brain waves and the oscillatory behavior just continued to develop with these much larger coordinated firing patterns as, later, I think they showed data up to 6 months. But it was just a really strong confirmation about how it was, in the astrocytes, they really organize and the neural activity. And it's actually really, you started to see the more, brain-like activity when the astrocytes reach that level of maturity. And so that, the implications were just really fascinating. But the bit about archaeolized, I think that was his term, basically taking Neanderthal genes and getting organoids with them. That was really bizarre. But these aren't. There's no vasculature in these organoids, so there was some discussion about how so that sort of limits their growth because it becomes nutrient limiting. But when two organoids come together and they touch, they start to fuse. And so there might be some way of mesh delivery and building up larger organoids with it, at least letting them fuse through some type of artificial vascular nutrient system. But there's just a lot of directions this work can go in, and it's just really fascinating stuff.
I
Can I
wasn't able to
actually ask you? Oh, sorry, beg your pardon. Please go.
go for it, dude.
Oh, Okay. I was only what I was going to ask was there was a part here where you mentioned the potency of progenitor cells turning into glia and then to astrocytes. That sounded really interesting to me, but I wondered if anyone here could explain that and if what that actually means in terms of significance. Is that like a big deal, or what is this special, or can anyone shed some light on that, please?
Well, I think that was just the differentiation path from the stem cells as the astrocytes were forming. Stem cells would the progenitor cells would differentiate, and that's how it's formed. But it's awesome that all of this works at all. It's amazing, because that's the developmental um, trajectory in these organoids is would certainly be our best model yet in terms of testing things, and if we can characterize what's actually going on and improve the methods of keeping and studying these organized, it could really really be fundamental.
Ah, see, thank you,
I
Suman.
was gonna ask. I was gonna ask. I didn't catch the talk. With when it comes to these the burst activity and the emergence of oscillations in the dish, was there a market difference between the dish with and without embodiment? Was there a big difference in
They,
the
yeah, they don't
go
have
ahead.
the data yet. They are still figuring out like troubleshooting, because when the robot moves, it kind of keeps destroying too much of the organoids. So they changed to have it basically a remote control that is wireless. But then there's a delay that is almost a second, I believe. So that's problematic for having like accurate movement and sensory information processing. But they are working on that part to like improve that. So they don't. I asked about that too, and they don't have that data yet because they are still working on making this work better.
That's interesting. That seems like it's calling for like a neuromorphic input, something that just really quickly low energy sends spikes, and you don't have a bunch of like delay for computation. But that's really cool. The reason I was asking is I was thinking about this idea of oscillations setting up in the dish, and I'm asking myself, does this add or detract from the idea that oscillations are the mechanism for cognition or computations related to cognition? There might be an argument for this just showing that maybe they are just byproducts of biophysical computations that are happening at a lower level. But if they did have data that showed interesting differences between in these oscillations, with and without embodiment, with and without, engaging the network and learning and things, then maybe there would be something to say to argue for the causal role of these oscillations. But yeah, really, really interesting research. I wish I had caught the talk.
Well, yeah, they
Yeah.
because they are. Oh, go ahead, Katerina.
No, no, you go first and then I'll.
Well, because we started. I mean, the chimeric organoids work, because they'll just fuse, and so there was some discussion about parts of, um, or differentiation into visual regions or some type of other sensory regions, and perhaps fusing them, and but they are at least thinking and moving in the direction of trying to understand whether, if you provide external sensory input, is that going to be driving in a more will that influence the development other than these organoids that are just sitting there in the dish, and the embodiment thing. Yeah, they're still trying to just get the mechanical placement and the connections stable and very early on, but
Yeah,
just
that's what I was going to ask about. You were saying that the robot was destroying the organoids, so I was just wondering how that worked, if it was a mechanical problem or an
Yeah,
electrical problem, or
it's mechanical, but I wonder why the robot can't be just be one. It doesn't. Does it really still have to be a physical robot? Can it be something that walks around in a virtual reality and have simulated visual and texture input? So basically, you tell the robot, like you tell the sensor what it's supposed to, what the input's supposed to be. Can you just do that instead? Like just say you're touching grass and make the sensor think it's touching grass so you don't have that physical mechanical stress. So that's one thing.
yeah, the way the
Maybe
sorry, go ahead.
That's clever. That's clever. Yeah.
yeah, yeah, and then I have another point, but go ahead.
Well, I was just going to say that's what Friston's group did. They embodied a dish in a game of Pong, and so the environment was mapped to the dish. So there was like a visual area, quote unquote, visual area where it mapped all the objects in the game to there, and then there was an area for motor control and all this, and it didn't require a robot. My other question was why can't the connections be mechanically fixed? I was just curious why that was a challenge.
yeah. I just think it's just the it's just they're that early on in the experimental setup, and they just hadn't refined it. Maybe that's why he was willing to talk about it. If it worked and the little robots were running around, it'd be he'd probably not say anything till the Nature paper. But on this question though, Katerina, of can you just do a simulation? I think about this a lot because part of in development in the day job, we start out with an all digital, and we have to really get high fidelity aerodynamics and control actuation loops and actually tuning an autopilot to work at those speeds. But as you go into hardware in the loop, and you actually there's a lot of surprises and retunings, and actually having to operate in hard real time is a different thing. Like you can't just go off and compute something until you're done and come back, because nature will just move on without you. And
But
so
just
actually, huh?
I'm sorry. But just to train or to see if there are differences, couldn't you just record 5 minutes of a robot running around and all the sensor information and then replay it to the organoids just to see if sensory input make a difference? You know what I mean?
Yeah, you can, but the lessons learned in the fine motor control will be different lessons. So you can it won't necessarily mean that if they learn how to play in the metaverse that they're suddenly going to be able to operate a real robot in nature. There's different tunings, and it's a difference. Um, they may be better at it, but they're still it's not the same.
Yeah, yeah, I see. I was thinking, you just record 5 minutes walking through a building and somebody coming out of in the hallway, and then you kind of give all that sensor information to the organoid and you see what the organoid would do in that moment, and then you replay it back to the robot. Um, so for the organoids, it's in real time,
But
but you
for
don't
example,
have the
to learn,
physical distress.
yeah, for example, to learn not to fall down, it's very there's some very high end physics to get that right. Um,
Yeah, in the future, but until they get to, I don't mean to completely replace it, but I'm just curious for now to know if that sensory information makes a difference in the oscillations and in the development of the organoid and the complexity and astrocyte, if they become more, if they replicate more. Like for that, I think that might be enough. And in the meantime, they would need to work on to do, for the future to do to have like real-time
Well, it's
processing.
possible, but say, for example, that these oscillations are control structures and that the specific timing of the real experience is um, is is what's actually driving the oscillations, and the actual metabolism and the spatial distributions and the different gap junctions support particular waves at particular frequencies. If those are actually what's coupled into the control system, it may be completely different in a simulated environment. And so it I mean, I do believe you can go far with simulation, but it would just depend on the nature of control, because we know these aren't von Neumann type architectures. These are, so they're more akin to they're analog devices in an instance. So the frequency response actually may be very directly coupled to the actuation consequences.
I agree with both of you. I think there is a big difference between virtual embodiment and actual embodiment. But I think, Katerina, you're right that this could be a good stepping stone in between the two, because there's just a lot of stuff to work out in terms of getting input in and getting certain behaviors out of the dish like this. And so maybe trying something in between, especially if they're struggling with the mechanical setup for the experiment. Maybe this would be a good place to kind of make progress while they're working on that.
So in order to get the delay out of the way through the wireless connections, they would need to build a quantum network. So we would need to hook them up with the guest invited speaker that we had a while ago that builds this quantum wireless internet connections, because
Well,
then you don't have
well,
delay.
why would you need quantum? I mean, what
Isn't
quantum
that
effects would you need?
immediately because you have this teleportation effect? So or so you basically if um, the if information processing in one
Yeah,
photon
the entanglement.
changes,
Well,
you have immediate response, so you don't have that second delay. You have an
well, I think
immediate.
the delay. I mean, a delay of a second is just a bad setup. I mean, you can have real hardware in the loop that, as long as you're rate matched, and since these are actual organoids, I mean, they could have an electronic system to address those delays. I think they're just so early on, but that's far more in the engineering than the science, though. And getting a good coupling sensory feedback loop without delays.
Yeah, but it has to be wireless, right? So you don't, because you have to have, so until now you still have to have tiny physical electrons. It's not like you put a patch on the brain. You don't have anything to hook it up to, right? And when we do that, let's say in mouse skulls or something, we always attach it somehow still to the skull, but you don't have the skull in organoid. So once it slightly moves, you completely screw up the hookup basically to have precise recordings from the same exact spot.
Yeah, but I mean,
Was that the issue? Organoids sliding around in the
Yeah,
dish?
they
Is
said
that the
then the
problem?
electrodes start moving and then it screws up everything, you know. And so it has to be
I see.
wireless
Maybe
and
they,
then yeah.
yeah, maybe they should check out what Friston's doing with the brain dish stuff, because they have like a really solid worked out stimulation recording
Yeah.
system at the
The
bottom of the
thing
dish.
is So the thing is, these organoids at this size, they cannot be attached to a dish. They have to be floating, and the floating has to be kind of constantly moving. So there is like there has to be a rotation at a certain pace. So there is enough solution penetrating throughout the organoid. And even then, if they do that, they still have in the middle tissue that after a while starts dying. Like I think they managed up to 3 years, but still, even with this flotation and rotation, they have tissue in the middle that doesn't get enough nutrient, and probably the pH level changes. I guess the biggest thing is the pH level, which leads me to the second thing. We asked why can't they, since Chimera worked pretty well, I said, why can't they build, and I looked it up again. There's a pretty established and pretty good way to make a cell-free artificial printed vascularization system and line it with lung cells that are there for gas exchange and pH level adjustments. So I think if you would 3D print it, you wouldn't have that issue, and then you wouldn't need to have them floating, and then you could have them fixed, and then you could solve also the electrode fixing problem. So I think they need to start making this artificial vascular. It's basically some collagen and some other stuff mixed that you can 3D print, and then you add the lung cells. That's my theory. I would add these lung cells. I never
See, I
tried it,
think,
but
yeah, they really need to get the vascular right because they're growing them to the point of it's nutrient limited. But they already know that two of them will fuse. And so, for example, like you were saying, if you could 3D print, say, here it comes a matrix of, this artificial vasculature and start the organoids in their little cells. As they grow, they'll fuse, and you'll have this whole brain in the matrix that's vascularized. But it would also be stable, and it would solve the frame fixing problem as well.
yep,
And
it's one of the biggest problems right now to 3D to vascularize artificial organs. But you can have because the cell one is really hard. Like people are working on this, companies everywhere around the world, and it's not working pretty well. But I saw a book actually that I looked into while moving. Anyway, I looked into that, started this in the beginning of the 2000s or even early to do a cell-free one, and that works actually pretty well, and it doesn't collapse as much. So if you could add in little mini pores with the lung cells, that would be really cool. He said he would look into the lung cell part, but I didn't have like I didn't look up the cell-free vascularization 3D printing method yet, so I would have to
well,
write him an
well,
email.
damn, Katerina, why don't we just do
I know, right? We should.
it?
I wish we could.
How much would that cost? Just to have a small setup lab on that, couple $100,000.
I don't know. We should look in, or like, because they'd probably give us, the starter kits or the samples, or the little organoids, so we could. But the 3D printing setup, Jean, you're familiar with how to do that, Katerina?
Well, there is an easy, cheap version. I don't know if that is good enough. That is basically a bunch of syringes and that you squeeze out noodles that you can make in all kinds of shapes you want and tiny as you want with a hole in the middle. That is pretty cheap and straightforward and not like, the company I work for, we have this, engineer, self-made complicated 3D printer that's really hard to calibrate. Like the engineers have to basically use it, but the easy one, the pretty straightforward version is just you have a bunch of syringes that you basically squeeze at the specific ratio into whatever you want, into discs, little noodles, whatever you want, tiny ones. And if that would be enough, And that's pretty stable. Like if you mix it with collagen and stuff, those noodles that come out are pretty stable. So we would just need to, make maybe, maybe you could even use Hamilton syringes. That, yeah. Anyways, I think there's
Stable
a cheap
noodles.
version.
Stable noodles. I love it. Stable noodles. And this isn't going to be one of those dried packets where you just add water and mix and leave in the fridge overnight, right? And you've got a jar full of organoids after that.
Well, it's really you mix collagen stuff, you mix in some cells, and then you squeeze it out, and you make little molds and you squeeze it out however you want.
Well, because that
Um,
really seemed to be their current limitation is they just can't, because the organoids will grow past the point where they can't really feed themselves well. And so they have to have them rotating just to get nutrients to, but that's, if they could solve, or somebody could solve the vascular issue.
yeah,
Of,
I could just go over there. They would hire me for a couple of weeks or so. I could go over there and squeeze them some collagen, mix lung cells, noodles, and then they could build their organoids around it.
or
Yeah.
let's put in a grant.
Yeah, I mean, it should work. I don't see why it wouldn't work. It's pretty.
Is
And then yeah,
sorry,
go ahead.
I have a question. Is part of the issue the permeability
Yeah,
of
exactly. Yeah, because there's no pores basically in between that let the solution in and the nutrient and the pH leveling solution.
Right, the volume increases faster than the surface, and there's no vasculature, so there's no way to feed, the cells, in the center, or it gets too big, and the diffusion can't keep up with the demand, and so that limits them. But if there was vasculature, especially if the astrocytes could actually reach the blood, because they're the ones that are going to be taking stuff out of the blood or whatever fluid is there and feeding the neurons with it. So if they were compatible with these noodles, that could work. That would overcome that, in principle, overcome that limitation, and they could grow larger.
maybe a way to limit the growth too, so you keep the ratio proper for saturation. So you don't have like the dinosaur or not the dinosaur, like why do we don't have enormous insects, So there's got to be a golden ratio of permeability
I
and
for one am
saturation.
glad we don't have giant insects.
Well,
And you don't want to ride a butterfly from Miami to LA. Okay, fine.
Well, that I could do in the backyard.
well, yeah.
In that little cabin. Especially if it's just a cell-free version first. I could, that's just collagen and stuff
Well,
like that. There's no,
yeah.
yeah.
So that would be the test. If we do the collagen, will astrocytes actually take to it? Will they be able to interface with it and draw nutrients out of it? That would be the key
Yeah,
step.
it's just
or,
collagen mixed with
sorry,
astrocytes. Yeah, that wouldn't,
or some kind
well,
of way to increase the saturation point or decrease the saturation point, increase the ability of the solution to permeate on a chemical level, if not physical.
Well, if the astrocytes were in the medium, that the only real source of enduring nutrition would be in these collagen noodles. If they continue to grow, that would show that they're actually drawing nutrients from the collagen noodles. And if we could feed them, you could do the controls where you have it nutrient rich and get growth rates versus nutrient poor, and what's gating them. That would be really cool.
It would. So we could have like penne and fusilli and
Yeah.
rigatone.
Yeah, we get characteristic different oscillations, whether it's penny or C.
Y'all making me
The in
Go to
hungry.
the
Portugal.
interesting thing is that it would be cool if it would be the snail shape, the spiraling
Oh,
shape of the organoid then,
what is that?
to make it look
Archimedes
pretty.
spiral.
Yeah, and then have different fluorescent markers, along the way, so it would look like some colored
Is
spiral
this
that
turning
would
into
look
an art
pretty.
project?
Right, yeah, different art.
Well, we want a nature paper. We need basically an art
Pretty
project at the
pictures,
same
pretty
time.
pictures.
The spiral, though, the permeability decreases at the center, though, maybe. So
People are still trying to
open
get
spiral.
cochlear hair cells to grow in a dish, so we could just do it on a spiral.
Yep.
Oh, there you go.
Well, we have a plan. Let's write a grant. No, seriously, we should write a grant and then
Yeah, we should. Let's do
just
that. If
do
you
it.
already, yeah, because this is a good approach for that. And that's one of their big problems. They'd probably want to go on it with us too.
Well, if
That'd be
you
cool.
solve that, every single organ growing company, even the nipples guy that is here on Clubhouse. Did you ever hear the nipples
No.
guy who makes artificial nipples?
I have
So
not
for
met the nipples guy.
okay, that was pretty in the beginning of Clubhouse. There was this nipples guy. He has this company in Israel. Oh, I'm sorry, we are moving. Stuff fell down. So the nipples guy, he so having for people that had to, because of cancer or any reason, reconstruct the breast, the nipple is still pretty hard to do, and usually it's kind of really ugly, sometimes made nipple, and he's an artist, so they hired him or made a company with him to basically artfully make this artificial nipple. And what they try is to have the tissue grow around that artificial one, so that it will become an actual nipple. And then the printed stuff kind of dissolves over time. You have different materials that would dissolve over time. But then the problem is that, first of all, the nipple will stay in place and also have that form, but then also to vascularize it, so that it won't die off. So there are a lot of companies, just a nipple one, because he was on Clubhouse here all
Of
the
all the
time
short,
talking
of
about
all the applications on a short list, Katerina, I'm impressed that you thought of the nipple guy. But
Yeah, you've taken us
no, really
from
though,
nipples to noodles. It's great.
nipples to noodles.
yeah, funny. But I don't know,
Wait,
he was
but
here all the time and presenting his nipple company. That's
well,
why.
that's. I mean, it's legit, but it does sort of
That
bring
sounds
a
like
smile.
that sounds like a really clickbaity room title.
Play with the nipple guy. I mean, that is fantastic. It's one of those things that unless it's in your circle of, experience, you don't know that people are suffering
Mhm.
from. It's fantastic. I love
Yeah,
to
no, it's
hear that.
there's it's a legit thing, but it is kind of amusing.
Yeah,
But seriously, Katerina, we should like put a grant in. This is
Oh,
obviously
yeah.
a general thing.
Yeah. Okay. Yeah, definitely.
You heard
So,
it here on Clubhouse on Science Society.
I heard Dennis ask about how much it would cost, so I'm just curious if you're the secret funder, maybe, Dennis.
yeah, if you know somebody with a lot of money,
I
I
wish.
can put the budget together. Like a cheap one. Well, cheap, as far as it goes to buy human cells and have a sterile
Hmm.
setup and collagen. But it's comparable. It's not millions of dollars to do this. It's less than. I mean, sure, if we could just rent a lab space, because I wouldn't want to give this to anyone just away. So if we could rent our own lab space, would be the rent of it, and then buy like a bunch of cells and some collagen and other stuff. It'd be a few $1,000s, and the rental, and us like working on this.
Well,
That's it.
yeah, it's
It's
not
not.
like it's going to be a one and done thing. We'd refine it and characterize it, and there'd be series of papers and stuff.
Yeah, send out the tissue. We don't have to have everything. We can send out tissue for sequencing and functional analysis to companies,
Hmm.
but it's like for sure, under 500,000, I would say.
So the purpose is our new purpose is vascularization of tissue, such as organoids.
Well, there are a couple of things though. In order to scale these up, there's needs to be a nutrient delivery system that scales with it, but also in terms of embodiment, having a frame, a rigid or stable frame that they can grow around, um, it allows the at least stable placement of sensory inputs, so they don't. But yeah, if you're just, I mean, if these organoids can grow around a lattice or that feeds, that can deliver nutrients. Um, what else is the angle with the astrocytes is they control the blood flow too with vasodilators and vasoconstrictors, and so in regions of activity they can increase or decrease. I wonder if these collagen noodles would have that kind of um, properties that
Well,
that the astrocytes could.
what it sounds like you're describing reminds me of the vascular system of plants, like the xylem and phloem and the cellulose structure. So, because I'm thinking, wow, all we need, all your, it's like people are trying to replace a living organism. But that's kind of maybe, speaking of chimera, maybe a plant that already is having its own uptake and respiration
Well,
could.
we are. I mean, these are living. These organoids are alive.
Right,
They
but I
just.
mean, grow them
Mhm.
inside of a plant of another structure that was plant-like, is what I'm saying.
Oh, I see what you're saying. Well, with the in the in the mammalian brain, it's the astrocytes really for vasodilators. There's there's nitrous oxide is one, but then there's another I can't remember. I'm kicking myself. But then the vasoconstrictor is the arachidonic acid, which is the endogenous ligand for cannabinoid receptors, which I thought was kind of cool. But they have a way to increase or decrease the blood flow, and so I wonder if how we'd pull that one off. But then they could actually start producing increased activity where they need whenever they decide that there's something that needs to be processed here. They can pull more nutrients out of the vasculature. This would be really cool. We got to do this. This is going to be fun.
Yep, that would be really cool. So shall we move on to the last one? Was it? No, last two. We still have two. Oh my god, we're almost as long as the. Yeah, we had then Dr. MD PhD, still MD PhD student, but she's quite accomplished. Dr. Gabrielle Zuniga. She is in at the South Texas Medical in the South Texas Medical Scientist Training Program and this MD PhD program in the third year. And yeah, she had this. She published this really interesting paper recently where she looked at tau-induced deficits in nonsense mediated RNA decay and how this decay contributes to neurodegeneration. So this was a really great presentation where she showed her research in Drosophila and how she used the tau transgenic Drosophila to recapitulate like key features of human tautopies, and then she showed the neural invaginations, how they are present in Drosophila and the human tautopathy. And then she went on and looked at the hypothesis that RNA is the information messenger for this invagination process, and nuclear invagination cause aberrant nucleocytoplasmic transportation of RNA, and how this RNA accumulates within the nuclear invaginations in tau transgenic Drosophila. She showed that, and that without these RNA accumulations, how these changes, how the changes in this RNA accumulations changes the invaginations in this tau transgenic Drosophila. And then they analyzed why might this excess in RNA export to be toxic to the cell. And they looked at this nonsense mediated mRNA decay, NMD, and that this is reduced in this tau transgenic Drosophila. And that basically NMD is this key mechanisms involved in that. And if these tau-induced deficits in NMD, they looked if this contributes to neurodegeneration, and what the potential link is between the blunted NMD and tau-induced invaginations. They showed that next, and how the transcription, the NMD target transcription within the nuclear invaginations, and how they are differently translated into protein. So it was a really thorough research of that mechanism, and it's really important work to know the exact mechanism in tautopy and how it changes in aging. That will hopefully give us, new targets to address that, and hopefully earlier on, so that neurodegeneration doesn't continue to do its damage. So I think it was really interesting, and she did a great job in presenting a really complicated data set. No comments? I did such a great job explaining this. Okay. Then on Friday, that was the last one from this week, was Dr. Azagun Henry. He just he's back at MIT. He is, well, he did his master's degree at Georgia Tech. No, his bachelor degree in Florida University, and his PhD in mechanical engineering at MIT. And then he did postdocs, and he explained, like, if you want to hear his story of how he became who he is now in his science career, please listen to the replay. He gave us a really detailed description from what motivated him already in high school, how the teachers were that motivated him to go into science, and to like satisfy his curiosity, and then also how his career went on later on. Yeah, I think that was a really important and interesting part. And Victoria, the team, you guys did an amazing job in interviewing him and getting that basically for us, that information for us. I think it's always really interesting. And Yeah, he develops novel energy system concept and also storing of energy from renewable energies, including solar energy, storage and transportation. And um, he uses high temperature concentrated solar power using liquid metals or molten salts and ceramic refractory base fluid handling infrastructures. And he creates high temperature thermochemical engineering conversion and reactor design. He works on methane pyrolysis for hydrogen production, solar fuels, direct contact exchange heaters. Like he works on so many different things, and he gave us a pretty broad overview of the research he's working on. And he also recently started a company that especially works on solutions for renewable energy storage facilities that are scalable. So so we can transition to that actually cheaper energy, and also good for preventing further climate change effects. So I don't know, Serena, if everyone else wants to pitch in.
He definitely did a good job to make mathematics sound really cool and really, really exciting and effective. I really loved the promotion he had with that. I wanted to do maths right after the talk. During the talk, I wanted to do maths.
Yeah, no, this was a really cool body of work. It was a real feat of engineering as well as science. I mean, the way it is, he is going for a utility scale means of storage of energy from solar fields. And it was cool how he said they imagine this huge vat of graphite containing the molten tin, and as it's pumped out, it's glowing, white hot. And all around those pipes, there's the thermophotovoltaic and that's just wrapped in this gold reflective foil to reflect any light that's not absorbed back into the molten tin. And so he keeps this huge vat of the molten tin in this graphite. It's about 2400 Celsius. He keeps it at. But then, during the pumping and the cooling, it, will get down to about 1700 and pumps back in. Um, but you can, if he just shut everything off, this thing is so big, it would take, months for it to cool. And, that's the economy of scale and going for the utility is that, the volume, it's that surface to volume thing again. um, but I asked about the footprint of this thing relative to the size of the solar field that would have to support it, and it was just very small. So you wouldn't even notice. It's still the solar field is the big footprint in this tower with this vat of molten tin. Would allow you 24/7 to recover, and the big breakthrough was the 40% efficiency on that, which is much higher than we had before. So it really does look like a big advance for supporting the energy in these big solar fields and anywhere else you needed to. I thought it'd be cool if there was like a family farm scale, but it's a sort of a diminishing return if you go too small on it. But at the large scale, it's looks like it's really promising. So it was pretty cool.
Yeah, I totally agree, and I hope that he becomes really successful with that, and yeah, and it will be used in the real world. So yeah, I'm wishing him all the best. We did, right? As we do. Wish him
Yeah, I
all the
agree.
fun he
I
can.
really appreciated that he's driven not only by his motivation, but by his desire to make a positive change in the world. And it made me think of how we are, we just owe so much of a debt to people who are able to take on the work that he's taking on and to secure funding for that. I just, excuse me, I just felt, sorry, choking on my tea. Yeah, it's just so much gratitude for that he followed his path.
It's also notable that there was a particular humility there when he said that anyone who looks at heat exchangers is not going to be impressed because it's it just he was in essence reflecting that it wasn't necessarily sexy, but it's just in a really important problem to scale up. And so that's to the point you were making, Victoria. It's he is going through the work to scale this up to where we could see it on a utility scale and draw the benefits from it. So that's really notable.
Yeah, Robin in the audience asks, "How do you pump molten tin?" And I'm thinking, you're saying it's not wasn't really sexy, or he was saying that, and I was just the whole time I couldn't stop visualizing that
Oh yeah, I mean
molten
his whole,
metal, working in yeah, it's
yeah, his whole setup was kind of badass, I thought. But um, he did go into a lot of the work was figuring out how to it was engineering of nature, is figuring out, okay, they're going to go with graphite, and then how do we get this pump system going, and what's going to be stable at 2400 Celsius, and and how do we do this? So there was a lot of the work, just getting to the point where you could actually build these facilities that he kind of glossed over. But yeah, no, it is really cool that because it replaces the eutectic salts that the concentrated solar panels use now, is just to be able to do it with liquid metal. And tin is cheap, and it's but being able to keep it in this liquid form, and I mean, metallurgy is really cool. Like, I remember opening a book on metallurgy, and like you do your random read, and at 900 Celsius nickel makes a fine solvent. And I'm like, are you kidding me? But yeah, when you get into these high temperature metallurgical applications, it's really crazy. It's just so far beyond your day-to-day experience. um, but yeah, he's
yeah,
got pumps of molten tin at 2400 Celsius.
yeah, that makes
Oh,
a
he also
fine
said when you
solvent,
look at it, it's still shiny.
and you can look at it. I mean, he must be wearing protective goggles.
Well, yeah, but
I'm just thinking of the
yeah,
light and
it's still
it still
shiny.
shines, right?
Yeah, he did say there was no indication that it was the temperature that it was. It looked, um, like, oh, that would be okay. Didn't it? Wasn't it Serena, right? He said there was no visible difference in it. It still was the shiny way it looked, at these super, super high temperatures. It wouldn't glow specially or bubble or anything like that.
Well, it was glowing, but it does glow. I mean, depending on the temperature, it's like his photovoltaic was in the infrared, but he said it was just glowing white hot. But if you take I think he had a YouTube video where they, because this whole you don't want oxygen around, so it's in this warehouse with argon atmosphere or something, because you'll start oxidizing the graphite eventually. And so you got to go in with protective equipment, because you need to bring in your own oxygen and make sure it doesn't escape. But yeah, it's just glowing, shiny molten tin. Kind of crazy.
Well, something else. Speaking of tin and cheap or not, is the sustainability of this and how we can get to a point. Hey, Sissy Rahim, here you come. Yeah, how can we get to a point of maintaining sustainability? Because I'm
Thanks,
thinking
Miriam.
the the shortages, because there is more demand for batteries, but also, why is it cheap? Whose land are we exploiting, and whose people are we not taking, the companies, not us, but taking good enough care of, that and are willing to work for certain wages that are, maintaining that low price, which might not reflect something reasonable for maintaining life, as with respect and kindness. So recycling is, I'm just wondering how much
Yeah,
can be carried out with recycling what we've already mined.
it's a good point. And I mean, tin's not a rare earth. I'm not sure what the mining chain is for tin and where it's mined, and but it's it's in solder. That's what solder mostly is, and so it is a readily available and very common element. But your points, yeah, it's a good point. Like the choice of metals should matter, and we need to consider the whole supply chain, especially on these utility scale
Yeah,
activities.
and I have to say also, if I just may keep on this for one moment, the ocean floor is at risk for mining, and it's not pretty. It's not. It's terrible. It's a really terrifying prospect. The legality of it, considering who has sovereignty, or not sovereignty, but who has who can take control over the ocean floor and intercontinental areas. It's really something to read up on, because the ocean needs our protection, and so everything living in there. Imagine tearing up the ocean floor, because there are manganese and just tons of things down there that have been deposited, and yeah. So like, there's, even if we go offshore, that's also at risk. If we It's important to develop, use what we've already got, would be just so great.
And I think it's just going to be, I think we've done a lot of damage to land animal habitat. So if we keep going into the ocean, and even especially those deep sea animals that only live in those like areas of the ocean where humans don't usually explore, I think that would definitely cause a change in the ocean ecosystem, where some of these animals will be coming up higher up, and it would definitely mess with like the food chain and things like that. So I think we should probably leave things alone. So we already mess with the environment a lot. So yeah.
Well,
Yeah,
though
really.
it should be noted though that at least this author's method is an important part of making solar energy reliable and stable at scale, which is you know in a very good way undermining the fossil fuel industry, in the sense that part of why we haven't been able to get more out of solar energy is because of its the unreliability of having, we have a constant demand, but that the sun isn't always shining in the right way, or cloud cover and so forth. But being able to store the energy goes a huge step towards ultimately helping the environment. So yeah, these are, yeah.
Yeah, yeah. Sorry, go ahead.
Serena, can you send me that information about what you're talking about with the manufacturing of solar panels? Very interested in this. Um, something like, is it like 40% of the solar panels come from China? And then, so if we were to look, Victoria, I just really appreciate you bringing up the supply chains and the extraction of rare earth metals, and also the indigenous populations in the different countries. And we can't just think about just our own country and allow them to politicize issues and then allow other countries to uh, you know, go into other countries, put them into debt, take ports and airports. build a One Belt, One Road initiative and basically lay claim to the most resources around the world as possible. And if we were looking at where these Tesla cars are manufactured, well, they're manufactured in China. And a showroom was just built right next to the Uyghur internment camp. So we've got to look at a global scale when we look at supply chains and rare earth minerals and resources and how they're extracted, transported, used, manufactured into goods, and then sold to people, and not just focus on the good that is sold and think that that's going to solve problems without going to the original source of where these materials came from.
Yeah, well,
So,
that
oh,
speaks to, sorry, if I could just make this point, it speaks to why it's so important that we think cross-disciplinary, that this, Dr. Henry's work is phenomenally important and really life-changing, could be life-changing for all of us. And in support of that, everything else, how could we support that work? Who else is doing work that supports his work so that we are acting with that conscience that we're speaking of, but not to diminish the importance of his work at all. Just to keep that mind, to keep that our eye on the humanity that we're discussing as well.
yeah, these are all really critical points, and I agree, Kyle, with what you're saying. But just to be clear, I wasn't speaking of the manufacturing of solar panels. I was speaking of the method that's pinned to the top of the room here, and it was about energy storage thermally from from solar fields, and they using a concentrated solar because we want to, in essence, heat up a medium. In this case, the tin, and it's stored in graphite. So tin is not a rare earth per se, um, but it's so this method is in support of green energy in a sense, and it would allow the continued generation of electricity from these solar fields, even at night, and during weather and so forth. So there's certainly plenty more to do, but this goes a long way in making the green energy initiatives at scale more reliable. So it's a really good advance.
Thank you. Yeah, this is a great discussion and a very important one. And I agree, being multidisciplinary and looking at problems from a lot of different angles is really important. So we don't create more problems instead of solving, while solving one. I agree. We did that a lot in the past, and we should avoid that in the future and look at it in from more angles. So I appreciate the comments, and yeah, this was the week. I think it was a wonderful week. We had really amazing guest speakers and discussions. So I appreciate everyone for coming and asking great questions and making our guest speakers feel welcomed and appreciated. Really a big thanks to this wonderful team we have here that came together and loves science and loves this club. And, we couldn't do anything without this wonderful group. So thank you, everyone, especially Serena, Victoria, Jamie, Dennis, Wisdom, Cecilia, Frank, Eric, everyone that's not here also right now. Uh, this group wouldn't be the same, and the guest speakers wouldn't be so happy to come, and I always get great feedback. What a wonderful group we are, how much they like coming, how much fun they had, how important the work is that we are doing. A lot of speakers say that we are doing something really important for science, and we are doing great. And so thank you for that, because that's very important feedback also. And I appreciate very much our group and what we do, and our guest speakers also appreciate it. And it's been wonderful so far. So thank you, everyone.
Thank you so much for making this possible, Katerina, and bringing us all together. We're following your light here, so thank you very much for that.
Yeah, thank you so much, and Katerina. It's amazing. I'm incredibly grateful you've taken the time and energy to put this together and this team together, and I'm really grateful for everyone that's taken part in your rooms. Learned a lot, so thank you so much.
Yeah, thank you. Thank you for coming and to support everyone also in the audience, people that keep coming back and engaging. And I think it's really important. We are doing really something, people, guest speakers said we are doing something really special for science and really different and really important. And I appreciate everyone that supports, that comes here. Um, we are creating something really unique and different perspective on science. I think a more personal one, because we also create, especially Victoria, started with this wonderful idea to interview in the beginning about the personal path towards a career in science, and how this project that we learn about came about. And it's such a unique path every time, and really interesting to learn about. So yeah, thank you for doing that. I would have never by myself came up with that. So, it's all a real team effort of everyone, and also all of our friends in the audience, because if nobody really shows up, it's okay. We still do what we do, but it also makes the speakers feel that people are interested in coming and interested in their research, what they do every day in their lab. So it's really important. So yeah, I wanted to take the time to thank everyone for that. And yeah.
Yeah, thank you.
And next week,
Go ahead.
go ahead.
I just want to say before, thank you. It sounds like you're going to launch into what's coming up next week, which is super exciting. If I can just interject, thank you so much for what you said. And I really believe that every one of us has our own unique attribute or whatever it is that we contribute, and that extends down to every single person who's in the audience, because it's so special for the guest speakers to come and see that people are here and really interested. And if there's one thing that I hope people take away, whether or not anyone's understanding the research or not. I mean, I have to go and do mad Googling before most of the talks to get up to some kind of speed. But I would hope that everybody takes away that just that they were in the presence of people following their own curiosity, and that's what people notice here is really the enthusiasm toward following our innate curiosity, because that's what gets everyone to where they are, who's coming here and presenting. And I think that's really the big unique flavor that I sense in here, is that we're really honoring that curiosity, and it's just so damn exciting to me. So I hope that people are picking up on that as well. So thank you. What's happening?
Well said, Victoria. Well said. Well.
Yeah, and every single one person here in this group and the audience, yeah, I totally agree, brings in something really special. Serena, with her, I don't know, with her amazing knowledge of everything, that
Well, I'm just
like
curious too.
stuns me every
Just another
time.
curious character.
And Dr. Shah, that is not here, she always has this amazing questions. Like this, like it doesn't matter if it's theory, simulation, how black hole works, the newest ones, to ask a really interesting question and suggestion there from any medical or applied science projects. I don't know. Like I'm really humbled to be in this group, and then Jamie, the amount he learned in such a short time, and the questions he comes up with now. I mean, not being a scientist in the lab is like, yeah, everyone here. I'm sorry if I didn't point everyone out. It would take an hour. What I find special about everyone here, but yeah, we all contribute a very different way of thinking, and that's what makes this so great and special. And everyone has her own way of enthusiasm, and yeah, it makes it this great group, and I really appreciate it. So yeah.
This is an incredibly safe space for curiosity. It's not shamed here, it's encouraged, and that's what and I think everybody else, I would say, in the audience probably agree on that too, right? It's good to be curious. And if anything, all of these amazing speakers have taught us, be curious. Everything else will follow from that. Everything. Right, Victoria?
Yeah, I would say so. It's well put.
Okay, so in order to stay curious, we have tomorrow Dr. Hildebrand talking about sex differences in pain signaling processing, which is really important because we know that pain medicine doesn't work necessarily as well in females versus male, and this is very important work that will in the future hopefully address this in pain treatment. Then Dr. Balderston will talk about individualized targeting of TMS treatment. Another, especially for drug-resistant mental health disorders, TMS has become quite successful tools, new tool for treatment, and doing this in a more individualized way would increase the success rate. And he published a study about that, and we'll present it here. Then on Thursday we have two rooms. One about if massive gravitons are good dark matter candidate, and then in the evening we'll have Dr. Moses, who published this amazing BCI study where they could decode speech in paralyzed people in order for them to communicate with the environment. And so far, he will also present not published new data for the first time from his lab. So we have to think about if we are allowed through different rules, he's bound to record the room or not. So we're kind of thinking about what to do there. But will most likely be brand new, like the data that is published and really brand new data shared here the first time in public. And then on Friday, we'll have Dr. Lipora talking about the artificial 3D printed robot skin that feels similarly to a human with more information processing, which is really interesting and I think also really important in different ways. So that's the next week. If nothing changes with rescheduling, if something comes up, but so far that's the plan for next week. It will be really exciting again, And thank you all for coming. Enjoy the rest of your Sunday, or if you are wherever you are, maybe Monday morning. Yeah, thank you, everyone.
Thanks,
Thank you.
everyone.
Thank you very
Okay, we'll close the room in
much, everyone.
3,
Have a good
2,
night.
1. Bye.
Go Science!